Good morning, good afternoon, and good evening, everyone, and welcome to Keygene's webinar titled R&D as a Success Factor for Biologicals Industry. I am Mireia Ibanez. I'm Biologicals Business Lead at Keygene. I have been involved in biological research for more than 10 years, so I'm so happy to be presenting and moderating this webinar. Today, we will hear 4 interesting presentations that will illustrate the crucial role that R&D has for the success of the biologicals company and for the industry as such. Allow me to welcome and thank our speakers of today, Martijn Olde, Olde Beckhaus from Vitens, talking from the Netherlands, André Negreiros, Corteva, talking from Switzerland, Irene de Bruin, Koppert Biological Systems, talking as well from the Netherlands. And here in the studio with me, Arjen van Tunen and Maurizio Rocha from Keygene. And of course, allow me to welcome you and thanks. More than 220 people registered, so thanks for your interest. And as you can see in your screen, there's a space, there's a chat, so please leave there your questions, and we will take them over, a few of them after each presentation. And also by the end of the webinar, there will be a session of questions and answers. So thanks for posting your questions. So moving forward, let's start with the first presentation of today. It will be held by Arjen van Tunen, CEO of Keygene. Arjen has an extensive career covering leading roles both in academia and private sector. As a CEO of Keygene, Arjen will be sharing Keygene's view and commitment to apply and develop technology innovations for and with the biologicals industry. Thanks, Arjen.
Thank you very much, Mireia. Good morning, good afternoon, good evening to you all. The agricultural sector is preparing itself for the future. All of us working in this vital sector of our worldwide society are well aware of the challenges that our planet faces for the coming years. The projected increase of the population and climate change brings a tremendous pressure to our production systems and to those who are taking care of producing our foods, namely the farmers and the growers. For this future, the biologicals industry is providing a great opportunity to meet those worldwide challenges, and it can add to improvements of agriculture, enabling a more sustainable food production. In this exciting real-time evolution, research and development, R&D, plays an important role in building for short-term and long-term successes. Over the years, R&D has already played a crucial role for the industry, amongst others by providing scientific proof of the benefits of using all natural biologicals. R&D continues to play a key role as a differentiation factor for the biologicals companies. The talks by representatives of Vitens, Corteva and Koppert that we will hear today clearly show that R&D is a valuable resource and it actually has an impact all across their companies, from regulatory to marketing and from marketing to sales activities. At Keygene, we are committed to develop and apply technologies for crop innovations, together with and also for our partners for more than 30 years to date. Our approach for supporting R&D for and with the biologicals industry lays in our integrated availability of technologies and expertise and aims to support the industry in all processes and stages of product development and exploitation. We summarize our support in 3 words: screen, understand, and design. Let's start with screen. Selecting and characterizing candidate products in a robust and scientific sound way is a key step in product development. When done successfully, it allows for minimal downstream efforts and costs while increasing chances of success. At Keygene, we screen and characterize the efficacy of products using our unique combination of digital phenotyping, genomics, and data science capabilities, either in a 4-pot setup at the whole plant level or with many, many more samples at the small plantlet level. The next step is developing a thorough scientific understanding of how a biological product works. Deep understanding of the products is of key importance from both a research perspective, but also from a commercial point of view. Keygene uses multidisciplinary approaches from different genomics and data sciences, related data sciences, to advanced digital phenotyping to understand the mode of action of a particular biological. And this mode of action we want to understand from a crop, pathogen, and or rhizosphere level point of view. That brings us to the designing new or adapted biologicals part. The new knowledge of the current generation of biological products generated by R&D will pave the way to design the next generation of biologicals products and the development of technologies that will result in a more efficient use of our resources while developing those new biological products or improved biologicals products. So let's move forward in successfully developing and using biologicals for a sustainable agriculture of the future. At Keygene, we are ready to support and also to strengthen you. And therefore, I'm proud that a number of our partners are willing to share how collaborative R&D developed as a success factor for their business in biologicals. And I'm looking forward to hear from them today during this Keygene's webinar. Thank you for your attention.
Thanks. Thanks, Arjen, for illustrating and sharing with us Keygene's view and commitment. So I would like to refresh you a question. So you have mentioned, and Keygene has a long trajectory of more than almost 30 years in developing breakthrough innovation for breeding industry. So why did Keygene decide to use all its capability and know-how to support biologicals industry as well?
Yeah, thank you. That's a very interesting question. Well, during the 30 years of existence of Keygene, we built up a significant expertise around genomics and data sciences and phenomics relating to molecular breeding. And these technological tools that we have developed in the past years, which are often proprietary and IP protected, we think that, and we are convinced, and we are using them also in our biologicals work. And the next reason why we decided to enter into the biologicals development screen, understand and design strategy is that also we saw that many of our partners were not only breeding companies, but they are also active in developing new agrochemicals and also biologicals. So we wanted to co-move with our partners and our customers, and that was the second reason why we decided that we would move into the biologicals. So it's both relevant expertise developed during 30 years of molecular breeding and contacts with the partners and demands and questions and needs from our partners.
Thanks, really interesting. So maybe we can move to the next presentation of today. It will be given by Martijn, Business Development Manager, Recovery Resources at Vitens. Vitens is the largest drinking water producer in the Netherlands. So thanks, Martijn, for being here today. We are very curious to hear you. When you are ready, the stage is yours.
Yes, I'm ready. Thanks for the opportunity, Mireia. I'm very happy to be able to tell here what Vitens is working on. My name is Martijn Oldeweghuis. I'm Business Development Manager, as you already mentioned. Vitens is the largest drinking water company in the Netherlands. The presentation I'm going to keep is about circularity, about resource recovery, and about the ambition of Vitens to make a positive impact with byproducts from drinking water production. Let me see, how do I go to the next slide? Yes. So Vitens is the largest drinking water company in the Netherlands. It's a public company and it's owned by 5 provinces and more than 100 municipalities. We invest more than 160 million euros per year to fulfill the increasing water demand and to stay an innovative, sustainable water company. Vitens produces drinking water from groundwater. So clean and healthy soil is extremely important for high quality drinking water. Water is collected in collection areas owned by Vitens, and the water from the wells is purified to highest drinking water standards. Via Vitens distribution network, it is transported to our customers. At Vitens, we often make the joke that water is a byproduct because we produce more than 60,000 tons of natural byproducts per year. Substances like methane, iron oxide, lime, humic acids, they are recovered from the groundwater to produce high quality drinking water. More than 99% of the byproducts find an application in the industry, but that was not always the case. In 2011, when we started our valorization efforts in a dedicated valorization team, costs to landfill or incinerate our byproducts were more than 1.5 million euros per year. And by focusing on quality and development of new applications for these byproducts, we managed to create value and become cost neutral. As you can see, earnings are still increasing and we will triple our results in 2022. So we managed to create value and turn waste into valuable products. We went from a focus on cost reduction to a focus on value creation. But since 2020, we shifted our focus to making impact. So we not only want to have honest financial returns from our byproducts, but we also want to make a positive impact on our environment and on the sustainable development goals. As you may know, the use of pesticides and fertilizers in agriculture has a negative effect on groundwater and groundwater quality, and it forms a threat to affordable and clean drinking water. And in the Vitens area per year, 2,000 tons of crop protection chemicals are used and around 70,000 tons of fertilizer. Together with commercial partners, we Vitens, we want to develop products from our byproducts that contribute to the reduction of pesticides and fertilizers. And these products replace traditional synthetic chemistry-based products. And this way we can contribute to the farm-to-fork strategy of the European Union and help to build a regenerative agricultural sector. So our strategy is to develop products that find their application in agriculture and nature, from the ground into the soil. In 2030, Spanneberg, which is the largest groundwater-based drinking water production facility in Europe, will be in complete symbiosis with its environment. It will be 100% sustainable, and all byproducts will be applied in agriculture and in nature. So currently we have 3 main products that find their way to agriculture. In the northern part of the Netherlands, the drinking water has a brown color because of the humic acids present in the groundwater. And our customers, they want blue instead of brown water. And Humphy is the result of an innovative process to recover and valorize the product from decolorization of our drinking water. Humphy is marketed by our partner PHC Den Ouden as Vulvic 25. And it has Humphy has proven to be able to reduce the amount of chemicals required with 30 to 50% and make crops more stress tolerant. So the second product are lime pellets. Lime pellets from water softening are brought to the market by Agrifirm, Circal, which is a green circular soil improver. It is slow release lime. This is a slow release lime product that can replace traditional liming products from European mines. And Ferro is the latest product that we are working on. It's a natural nutrient, an iron nutrient that can replace traditional synthetic iron chelates. Chelating agents like EDTA can be found in Dutch groundwater and are a problem for drinking water companies because these compounds must be removed. In our own area, we are working together with the fruit sector on pear cultivation in the Betuwe. Pear cultivation requires high amounts of iron to get green pears instead of brown. This way, we offer farmers a natural circular alternative to protect our water resources and replace traditional iron chelates. And now something about the importance of R&D for Vitens. In general, R&D and piloting are very important for the valorization of byproducts. It gives the science-based evidence and the proof of concept that a product is doing what it's supposed to do. With this proof of concept, Vitens can attract commercial partners that can pick up further development and commercialization of the product. Before we commercialized Humphy, we have done several trials with partners on several crops to prove that treatment with Humphy gives higher yields. And we have just finalized a study that shows that the amount of herbicides can be reduced with 30 to 50% when blended with Humphy. With lime, we have done long trials on grass before AgriFirm got interested. For Ferro, we are finalizing the proof of concept. We have done several successful field trials on pears in the Betuwe in the Netherlands, but we have also done in-house trials with botany and Wageningen University on pak choi, basil, and soybean. Keygene did a transcriptomic study for us on pak choi plants with an iron deficiency. These plants were cultivated by botany in the Netherlands. This revealed the mode of action of Ferro inside the cells. So the conclusions of this study, the main conclusions from this study were that Ferro can be a natural alternative for synthetic iron chelates. Its mode of action is supplying bioavailable iron to the crop, comparable with the iron chelate. But Keygene also found indications that Ferro has a positive effect on root development and on nutrient transport and the secondary metabolism. This is probably due to the presence of fulvic acids in Ferro. And soon we will discuss with potential commercial partners how and where to commercialize Ferro to reduce the use of chemical iron chelates. If you're interested, please reach me on martijn.oldeweghuis@vitens.nl. Thank you for your attention.
Well, thank you, Martijn. At this moment in time, there are no questions from the audience. So I would like to stimulate all people from the audience to type in their questions on the chat. Then we will see them on the screen and I can forward them to the speakers. So Martijn, I have a question myself for you. You basically have two reasons, as I understood, to do this type of R&D on side products from your drinking water cleaning process. It's probably cleaning the ground itself, getting the removing or preventing that agrochemicals are getting into the soil or that too much nitrogen is coming back in your drinking water. On the other hand, you're also valorizing the products you let's say isolate from your drinking water process. So if you take a look in the future, what is going to be the most important aspect of that type of those two elements from your strategic R&D? Is it the valorization of the side products? Are you going to be a biologicals company? Or do you
our core business is providing clean drinking water to our customers. But of course, the quality of the soil is very important. So if we can use our byproducts to replace conventional synthetic products, then it's a win-win for us, because we valorize our byproducts, so we earn money with it. And next to that, we also prevent the pollution of our resources, our water sources.
Yeah. And I have another question. Those pairs, plus and minus the farrow contribution, how do they look like? Plus farrow and minus farrow, and what do the growers think of it? I mean, we have a lot of growers here in de Betuwe and in Gelderland. What do they think of it? Are they prepared to pay a premium, or how does it work?
It gives them the opportunity to become a more sustainable pear cultivation sector. So there's a label, Planet Proof, and if they use farrow instead of synthetic chelates, it gives them the ability to become a Planet Proof company.
Okay. And do you have plans, or are you already doing? Are you exporting this product, or?
No, not yet. Not yet. We are doing the last demonstration in the Betuwe at the moment, and if we are successful, then we are ready for commercialization in the Netherlands.
Well, I think it's a very intriguing, let's say, application. Indeed. I would like to wish you good luck with it, and lots of success.
Thank you, Arjen.
Thanks, Martin. Thanks, Martijn. It's impressive to see how Vitens, as a water company, is in the first row, coping with the impact that traditional agriculture has on our drinking water.
There's maybe one or two questions which are popping up from the audience. Maybe you can read them?
Yeah, so
there's a question: what is the balance, Martijn, you have to manage between IPR contracts and the required trust between the multidisciplinary R&D team members during the phase of R&D collaboration? That's from Marianne.
When IPR is intellectual property.
Yeah.
Yeah.
And
so can you repeat the question? How
do you manage the IPR contracts and the required trust between the multidisciplinary R&D team? So you are collaborating with botany and Keygene, and there is IPR involved. How do you manage that during this pre-phase of product development? Who owns the patents, for instance?
Well, we have patents, but we have patents on the process and the manufacturing of the product. And blending of the components to get the product, but we don't have IP on the product itself, how it works in the field. So that's really something for the company that gets involved.
And I guess that you own the product itself also. You don't allow, of course, Keygene and botany to bring the same product on the market. You have arrangements around that, or not?
Well, we are the only one that can provide a product. It's a unique product with unique properties, and the product is only available from drinking water from Spanneberg, which is one of our sites. So it's a very specific, unique product.
So there's the last question, if I may.
Yeah. Let's go.
So there is a question from the audience who say, well, it looks so easy from the slides. Why didn't anybody else do this before?
Specifically what? Developing farrow?
I think that's the farrow, yeah.
Because we are in a unique situation that we have humic acids in our drinking water and very high quality iron oxides. So if you combine that and if you invest in technology to make a product out of that, then you are the single source of the product. Is that answering the question? So I don't think anybody else can make the product we have because it's quite unique.
So it's a unique manufacturing and producing process, right?
Yeah, and the source is unique.
Okay. Yeah.
So Thanks, Martijn. We would like to ask you one last question.
Yes, Martijn. Good afternoon. Thanks for your presentation. While you were presenting, I was wondering if you have any idea of the market share of these products that Vitens has on the market with collaborators, and in the Netherlands, let's say, and if this is being, more and more applied in agriculture, which is a trend, and I believe that that's the case. If you see back that on the quality of water, so farmers using organic biological products rather than chemical products, and if you can follow that up on the quality of water that you monitor.
Okay, that's an interesting question. Thank you. So we don't have any market share yet, but we are aiming for, let's say, 50% of market share in the fruit sector in the Netherlands. That's a very ambitious plan. But we hope that we can replace 50% of the iron chelates specifically used in the fruit sector, for example, in the Betuwe, in our own area, and that way to have a positive effect on our groundwater quality. So we are pretty ambitious, but I think if the product proves that it works the way it should work, so we should be able to replace 50% of the synthetic chelates.
Yeah.
That's the aim.
Thanks, Martijn. Thanks for sharing this exciting experience with us today. So when we can move forward to the next presentation that will be held by André Negreiros, responsible for seed applied technology business in Corteva AgriScience Europe. Thanks, André, for presenting today. We are very excited to hear your experience and also Corteva experience in exploiting the whole potential that R&D has for keep building successes in biological. So when you are ready, go ahead.
Good. I hope you guys can hear me well and can also see my screen. Is that working?
Yeah.
Okay, so first of all, thank you, Mireia, the Keygene team for the opportunity. I think it's a nice story that we have here to share, and definitely something very new, So before I start, I also would like to say hi and good morning, good afternoon, and good evening to the whole audience that are from different places of the world. Thank you for your presence here. So as Mireia was saying, I'm responsible for the seed applied technology, so the seed treatment portfolio in Corteva. This is an area that we used to call here internally as a startup inside Corteva, because it's one of the youngest, I would say, portfolios that we have. So it more or less started together with the company when the company got together. And I'm in Corteva since 3.5 years. So I'm working in seed treatment since almost 10 years, but in Corteva since 3.5 years. And I joined here. And since I joined, I implemented this biological story. And this is more or less what we're going to share here in the few next slides and few extra minutes. So we're going to have some time for questions in the end. So if my connection fails in the end, it's because the question is too tough. So don't worry about that. So I'm just joking. So let me try to move my next slide here. So yeah, about Corteva and our portfolio. So I guess most of you have already heard about Corteva. So we are one multinational company present all over the world. We have a big portfolio of crop protection products, meaning conventional chemicals, but also biological products and seeds. Of course, we do also have other services like digital, like agronomy services, but we are, the majority of our business are crop protection and the seeds. So I try to split here in this slide all the three pillars to make it easy for you guys to understand where I really work and what kind of work that I do. So as I put here in the left side, you see a portfolio that is on the farm portfolio. And what I mean about on the farm portfolio, this is the portfolio of crop protection products. Again, it could be conventional chemical products, but also new biological solutions that are also crop protection, that are applied directly in the farm. So like foliar applications that the farmers do themselves at their crops at farm level. So there we also, at farm level, we do have, I would say, digital service and agronomy service that I put in this pillar of on-the-farm services. So on top of the slide, right in the right corner, I put in the seed, and this is is on seeds business. So Corteva is one of the major players in the seeds market, especially in the hybrid crops all over the world. And this is a very, very important pillar for our business here. And here in the bottom is the area where I'm responsible in Europe. And this I put on the seed, because this is really what my area do. I mean, we applied crop protection, again, conventional chemicals or biologicals into the seeds. And then the seed companies in general, not only Corteva, but other seed companies in the market, they use those products to treat their seeds and then deliver that to the farmers, okay? Just to give you an overview. I also add this slide here that goes really very much in line with Martin mentioning in his previous slide on sustainability. I'm not gonna go deep here, but if you have some time and interest, I would recommend you to check the sustainability.corteva.com, where we have all the 14 goals and commitments that Corteva has to achieve on sustainability goals by 2030. And the importance of biological to that. You know, when you go through every goal, you see that the majority of those goals has something related to biological. So the importance here for me is that, I mean, just to raise how biologicals is already important to us, even if we are a company that is operating in this space since 3 years, but it will be more and more important for the future. Moving forward, I want to go a little bit deep in this slide because this is a very important one. I think this is the way Corteva see biologicals, but I see that the industry in general is also seeing the market in the same way. So you have clearly two different segments today. One that we call biocontrol, and biocontrol could be biochemicals, could be pheromones, could be microbials, all from biological origin. So it could be alive microorganisms, it could be dead bacteria, could be product of fermentation. But the most important piece of these biocontrol pieces is that these have a biotic function of controlling pests in general. So here, when we talk about biocontrol, we say that we have a very specific regulation, well regulated as the conventional chemical products today. So it goes through a level 1107 regulation in EU, goes through registrations, lots of studies, dossiers, submissions. So very long years of time to get a product registered in the biocontrol space. And again, for biotic functions, it means for bio, as a bioinsecticide, for example, or a biofungicide, or a bionematicide, but something that has a relation with control. And then in the other side, we have, I would say, a new market that are the biostimulants. But then you're going to say, look, but biostimulants are not new. I mean, biostimulants are there since a very long while. This is true, but it was never as regulated as the biocontrol business or the chemical crop protection business. It's being, I mean, this started to be, I would say, more regulated now. We still do not have a harmonized regulation for the biostimulants yet in EU, but it should come soon. And this is, for me, the newest area where I see a very strong investment from the industry, in general, the crop protection industry, in this segment. Why? Because we are starting to see a clear benefit of those products in the crop protection industry. And what is the function of the biostimulants? As the name says, it helps to stimulate several different properties within the plants. But it is really focused not in controlling a specific pest, but more in the sense of the abiotic stress management. Meaning, what is abiotic stress? Potentially drought tolerance, or a cold stress tolerance, or stimulation of the growth of the roots, have a stronger plants, have faster emergence of the seeds. All of those effects could be included within this biostimulant, biofertilizer segment. Okay? So moving forward here, I have a slide where we have on commitment, and I think this is, again, this is the Corteva commitment within biologicals. But in general, I see the whole industry investing a lot on this area. Something that was not common, and it's turning to be more and more common now. I mean, I'm in the industry as the crop protection, conventional chemical industry since more than 15 years. So I work for several big players in this market, and I always observed how tricky was for those companies to work in this biological scenario. You know, biologicals, they are completely different than chemical conventional solutions. You know, a biological product, it could have 6, 7, 8, more than 10 mode of actions in one product. For a chemical, this was much easier. You know, you have a specific molecule, very well known, with a very specific mode of action, with a well-known performance, or a very black and white performance in several cases, like insecticide. You apply the product, the insect is there affected. With biologicals, it's a bit different. I mean, we have all the complexity beyond the mode of action, but the performance also, it's quite different, because, you know, it may vary a lot from soil to soil, from weather condition, from type of the species of different pests. So the importance of R&D in this segment, it's really, really high. So this is one area where I see the industry investing a lot of money, especially with the commitment that I put here to have in the end a product that it meets the efficacy that the farmers want to have. It's also very safe to the users of the product, to the farmer, to the environment, and something that it's reliable. So different than several solutions that were there in the past, where they were well known by not having a proper mode of action or proper ways of working. We are trying to bring something that has a proper script. So moving forward and going a little bit more deep on our current portfolio, I will talk about Lumidapt. Lumidapt was the first product we brought to this space that we call growth nutrition seed treatment. And since the first moment we test the product, we could clearly see the benefit of using it. You know, we see better rooting. We see clearly stronger plants, but we were always struggling to say why it would happen. As I said, with biologicals, with plenty of different mode of actions, we were always missing the reasons why we were facing this such good benefits, you know, and also in areas where we were not seeing a clear benefit, to explain why we are not seeing the benefit. So after that, it was more or less 3.5 years ago, we said, look, let's invest deep dive in R&D to understand the molecule, to understand the product. And then was when we come up meeting with Keygene, and together we developed over 1.5 years of studies within the impact, in the combination of impact with the conventional chemistries. And we were quite confident after that, with several answers that we never had before. So we were much more prepared to move forward and have a proper positioning of our product. So then after all this information, we put together, I would say, strong pack of R&D data. We provide with that pack trainings for our internal team. So we were able to train on teams, so not only on sales team, but on technical team to support the sales and the demand generation of those products in the field. And also to have a proper argumentary to, when we face questions coming from the farmers, we were prepared to give the proper answer. So with all those R&D information that we generate over this 1.5 years, we really had a very nice materials, videos, webinars to do those trainings. Huh? And here it's my last slide. I mean, the numbers talk by themselves, but with all this information in place, the whole team felt much more confident to approach the market. This is a product that today we have registered all over Europe, not only EU27, but non-EU countries, Russia, Ukraine, Turkey. So all the countries that we have here in our Corteva European headquarters. And in this last season, we achieved more than 10 million hectares of hybrid crops treated with Lumidapt in Europe. So I showed in a slide before, because now after Lumidapt, the success of Lumidapt, of course, we also face a pressure from the farmers, from our teams, to come with a new portfolio. We just launched during this winter cereals time. So now a new product that is Impact, this one it's behind me here. It's a product exclusive for the cereals crops. We already have the pre-launch during this season. And again, we have very strong expectations that we did grow quickly with Lumidapt. We are also expected to grow and have similar results with Impact. And our objective for the next 5 years is to launch a new bioproduct in seed treatment every year. So this is on goal, this is on pipeline today. And we are very much open, as Martin also mentioned in his presentations, to partnerships. When I mention here Lumidapt is not an internal Corteva solution. This is also a product of a partnership with another European company, R&D based company. So we are very, very much open to receive the contacts of those R&D companies that have an interesting product to together develop this in the European market. So thank you very much. And again, I'm open to questions.
Thanks, Andre. It was really exciting to see the trajectory you are taking into the biologicals and what role they can already play from the very beginning on the seed establishment. And so thanks. If we move to questions, maybe there's...
Yeah, first question, Andre. Thank you for your very nice presentation. But can you elucidate part of the mode of action of Lumidapt? Why does it give this better root performance? Is that from a technical point of view? Can you say a little bit about it, or is that still not really known yet?
Definitely, we can say Lumidapt. It's also based in as I was explaining before, 6 or 7 different mode of action. So this is a product that it's registered in EU as a EC fertilizer. And why? Because it's again, once we move treated seeds around EU, we choose the EC fertilizer route because we do have a harmonized legislation in this place that allow us to have liability to move treated seeds around. And then we claim most of the claims around the product is on the results of the nutrients absorptions that we have in the fields, or that we allow the plant to do in the fields with Lumidapt. So Lumidapt, it's based on FUVIC, humic kind of properties. So it's a IP protect active ingredient, also blended with micro and micronutrients.
Okay. There's a question from the audience, from Christophe for André. He is asking why Corteva is starting from zero, while they could also have been taken over one or two biologicals companies to start with.
It's a very good point. As I said, I said that we start from zero because, again, this business or this market, it was already there, but nobody until we did was able to really fast ramp up penetration in the market. So we decided, as I said, we decided to partner with a very reliable, small startup, but R&D based company that help us to have the product very quick. So this is not a Corteva product, as I said, a proprietary product. This is a product of a partnership, but we helped it to fast ramp up. We are working within our pipeline on proprietary solutions, proprietary products, but we are also looking after companies. So again, we are open to partnerships and to fast track solutions. So what is very important for me, it's really to be as a pioneer bringing those products to the market.
Okay, thanks. There's a question from Raju for André. Do you see the combination of biologicals with probably traditional chemicals as a more feasible approach for food security as well as environment protection?
This is an excellent question, and I missed that actually during my presentation. This is exactly the view that Corteva has on the balance of using conventional crop protection solutions together with biologicals. We do believe that this combination will really help to meet the challenges on sustainability that we have in EU and around the world. So definitely, yes, we do believe in using those two technologies together.
And probably a related question from Giovanni. Do you have products which replace chemical products, so which will not work synergistically with chemicals, but do you have biologicals with replacing chemicals? Do you see a market for that?
We do have today already biologicals. As I said, the biocontrol solutions, they are replacing chemical solutions in some cases. Does it work as efficient as a chemical solution? This is the key question. I think we are not there yet with the biologicals, with the biocontrol products, but I expect that in the future, with strong incentive, strong investment from R&D, we will get there. Today, we already have, let's say, biological products coming from fermentation of bacteria that are bioinsecticides registered for the organic production, like the spinosins. And those are products that are biologicals, they are organic, and they are replacing the chemical products. So for sure, I think that in the future, this can happen.
Okay. Well, another interesting question, I think. How do you find soil health? And how can you prove that your products are improving soil health? That's from an anonymous guest, but that's an interesting question, I think.
Yeah, this is a very, very interesting question, and it's a very important one. So what we see when we apply the product, and of course, when we do it in seed treatment, as you can imagine, the amount of product that we put in the soil, it's really, really minor. So it's really small. So of course, around the seed, so the soil around the seed that we have the bacteria there, we see an increase of this bacteria population. So we already did a study in the past measuring soil, a normal soil with the product applied in there, and the growth of the beneficial bacteria, beneficial to the plant, bacteria versus the potential negative strains of bacteria and fungus. So we have studies on that. As a seed treatment, to be completely clear, it's very hard to really see this benefit because the amount of product that we apply, it's quite small. But when you go to other scale of application, like we also do have a foliar application in furrow application, we can see the benefit in the soil. And this, of course, is only measured by laboratories. So you cannot clearly see that by naked eyes in the fields.
Okay, there are lots of questions. There's a question from Sheetal to André. Which are the top 3 innovations which you need, according to you, which will speed up the development of new biologicals and adapted biologicals? So she wants to learn 3 top innovations from you. What are your needs for R&D?
Very good question. So we definitely need, the key focus and the key gap today for our European markets are insecticides. So as you can imagine, since the ban of the neonicotinoids in EU, several other chemical products were also banned, especially in this insecticide segment. So innovations as a bioinsecticides will be crucial to keep our market, the farmers here, competitive versus the rest of the world. So this for me is by far the number 1. And then of course, all the other biocontrol products like biofungicides, bionematicides, that is also a new market, are very, very important. And the market is willing for that. And also biostimulants as the one that we brought in the market, like Lumidapt, where you can clearly see the benefit when you apply the product in the plant. For me, there was a lot of lack of credibility of several products in the past, because there was a lot of promises, millions and millions of extra tons, very high yield gain, and the farmers were not able to clearly see that. So I think bringing more R&D, more reliable claims behind those products will also help to develop this market.
Okay, we have 2 last questions, I think.
Go ahead.
Pawan, question to you, André. Do you have plans to offer a package of seeds and biologicals?
Yes,
to the farmers?
We are already doing that. We are already doing that.
Yeah. And there's also a question from Arnaud or Arnoud. Do you see any interaction between the bioproduct Lumidapt and various and different varieties, different crops probably?
It makes sense. As I said, for Lumidapt, for example, it was specifically developed, including the micronutrients in it, for hybrid crops. So most of our tests were done in corn, sunflower, in oilseed rape. But we do also have a lot of tests in soybeans that are ongoing. But we can clearly see that different crops respond differently to the same product. So this is why we are developing other products like we did with Impact for cereals to other crops like veggies. Veggies is one of the key challenges that we have, because we see this industry in Netherlands for that. It's very important. We're really looking after some new solution. And other hybrid crops like sugar beets, like field peas, like peas, soybeans, and then we know that those crops, they respond differently to different products. So we cannot use Lumidapt across all the crops. So we need to develop specific products for specific crops. And this is the direction that we are working. We're still not there yet. For those 3 crops we already have. We have now for cereals, but next year probably we're gonna launch our first product also for veggies and soybeans.
Do you understand that question?
So
there's a last question, I think, which I not really understand, but I will read it out. Maybe you can comment on it, André. So in the work you have been doing on the mode of action of biologicals, did you find that the crop derives a direct benefit, I presume, from the biological application itself? Or is it the microbial activity that the biological promotes? So if it's stimulating the microbial growth around the roots, or are the biologicals directly stimulating the crop itself? Or
it's an excellent question. I mean, I'm not sure if I'm that R&D deep knowledge to answer, but what I can say, based on the studies that I present, is that it's both. Actually, it's both. You know, as I said, the complexity of the mode of actions of the biostimulants or the biocontrols, you can have in a biostimulant product from different sources, like the algae-based biostimulants, or in this case, the humic fulvic acids, or the ones based on plant hormones. You can have 6, 7, 8 different mode of actions. So few of those mode of actions can definitely affect directly the plant. Others can affect the soil microbiome. And you know, I think in the end, both together are the ones giving the plant response that we are facing today.
Do you want to comment, Mauricio, maybe?
Well, I fully agree with what André is saying. I think once you apply a biological, as he said, you have so many different mode of actions that are possible for these biological products that you would need actually to look into that, right? So when you are looking at the effect of the biological on the plant and just looking at the phenotype of the plant and not understanding exactly what is going on, then you are not exactly sure on how, you know, that process, how that phenotype is coming so it's happening. So in that case, you would need to look and plan experiments projects in a specific ways to look specifically at different type of mode of actions that you could say, okay, we see that with, for instance, with Lumidapt, we have an increase of certain, as André said, beneficial bacteria that could be supporting the phenotype we see. And all those questions, I think, are very relevant for what also André said, that we probably need to move away from this, you know, snake oil mentality of biologicals that we had in the past. So, and the way forward is R&D.
So you have to take a look at the crop itself, at the roots from the plant itself, but also to the rhizosphere and maybe also to the microbials themselves, right?
Definitely. I think, of course, once you have a product, you already have more or less a guess of how, you know, that product should work, and you sort of understand and think, okay, this is what we are, you know, looking for, and this is the effect of the product. But definitely, when you are dealing with biologicals, is an interesting aspect is to look at indirect effects that can come with the application of these biologicals, and looking at microbial activity in the soil, functions that could be supporting the phenotype you are looking at, you are seeing. It's definitely something very important to do.
Thanks. Thanks, André, for your time. It was really, really nice to have you here. So, moving forward, last but not least, Irene, she is team lead microbials profiling at Koppert. And indeed, Irene will talk about a really important aspect, and it has been somehow anticipated, which is the registration requirements. So, thanks, Irene, for being here. When you are ready, you can
Thank you, Mireia. Can you hear me?
Yeah.
Good. Well, thank you for this opportunity to represent Koppert in this webinar. It's a very interesting webinar so far. So hello to everyone. So I'm Irene de Bruin. I'm the team lead of microbial profiling. I've started working for Koppert 2.5 years ago. And before that, I was working in academia and always focusing on biological control. And very recently, I became the team lead of microbial profiling, and I will describe further in my presentation what that involves. But first of all, I will, and I hope my slide is now changing. Yes. First of all, I'll just introduce Koppert. So Koppert Biological Systems is founded in the 60s. We have about 1,500 employees worldwide, and it's a family-based company. And it initially started as a company who produced a beneficial mite to control spider mite infestation. But now we've expanded our portfolio massively. So Jan Koppert, the founder of Koppert Biological Systems, he was basically a farmer. He was a dedicated cucumber grower. And of course, he had to control diseases and pests using chemicals. But the efficacy of the chemicals were decreasing, and he also started to develop an allergy against the chemicals. So he needed to find a new solution. And one of the solutions that he found was a spider mite that's so a predatory mite that could combat the spider mites. And now this is, he developed that into a product called Spidex Vital. And that's basically the start of Koppert Biological Systems. So Koppert is now the market leader in biological crop protection and also natural pollination. We really work and partner with nature, really try to find solutions from nature, and we really work for the growers. So we want to make the agriculture healthier, safer, more sustainable, and also more productive, of course. And now Koppert solutions are applied worldwide. So Koppert is producing parasitoids and mites and bumblebees, and that's still the majority of our products, but we also have microorganisms and nematodes. And I will mostly focus in this presentation on the microorganisms. So of course, this webinar is about research and development, and R&D is really an important factor in Koppert's activities, because our R&D departments are basically going into nature and looking for new insects, mites, but also microbes to find solutions for the farmers to control their diseases and pests. And like I say, we really work for the farmers. So we really look into the markets, we talk to the growers, and if they have problems, we try to find solutions for them and help them out. So in our microbiology department, we start with the microbial profiling. So that's the team that I'm leading. We are characterizing new microbes to see whether they fit in our processing facilities, and if they are effective. So in the microbial profiling team, we characterize the microbes. And then if we find interesting microbes, they will move on into our processing team, which perform the upstream processing. So basically upscaling the fermentation processes, but also investigating the downstream processing. So basically collecting the biomass and also formulation. So adding co-formulants to really form into a product. And we have several types of products, for example, liquid products, but also powdery products and more granular products. So our R&D department, the processing team, is really involved in the upscaling, but also in investigating the shelf life and ensuring the efficacy and stability of our products. So at Koppert, we really have an in-house policy. We really try to develop everything in-house. So we have our R&D department and also our production facilities, quality control, everything is in-house. So we have several types of products, which are gram-positive bacteria, gram-negative bacteria, several fungal products, which include entomopathogenic fungi, which are affecting insects, so killing insects, but also soil-based fungi, which are controlling the soil-borne diseases. And even though they're not really microbes, the entomopathogenic nematodes, they're also part of the microbiology department, basically because they're smaller than the insects, and therefore they're included in our department as well. So our R&D department is not functioning on its own. We really collaborate a lot with our, for example, our agronomical development department. They, of course, in the R&D department, we are developing prototypes. They need to be tested for efficacy in the fields. So first we start with efficacy in lab and climate room skills, then we go to greenhouse skills, and then we really go to field trials. We collaborate also a lot with production and quality assurance and quality control, because of course we need to know whether our processes really fit our production facilities. And basically in the center, which are really important for the whole development of products, is the market access. And that's basically our registration department, because all the processes we develop should really comply to the registration requirements. Before I really move into the registration requirements, I just want to also show you one of our products, this is our main microbial product, Trianum. It's based on fungus Trichoderma afroaceanum T22. So we have a powdered formulation, which is Trianum P, and a bit more granular formulation, Trianum G. So the Trianum P, you can apply it in irrigation systems as a spray or in furrow, while the Trianum G can be applied also in furrow or mixing with substrates. So here you already see that we try to adapt our applications to what is also requested by the farmers, and to make sure that we can apply the products in the machinery that most farmers have already and use for applying fertilizers or other chemicals. So here I want to show you a movie, and I hope it will start running soon. And this is a movie where we look at maize roots, and on the left is a treatment where there's no fusarium or trianum, so just a control treatment. In the middle is a fusarium treatment, so the soil was infested with fusarium, while on the right is fusarium treated with trianum. And I will start moving now, and I hope it will run. So you're looking at the roots. And now on the left, you will see, so in the control treatments, the roots stay nicely white, while in the middle, the roots are really browning. And of course, this is also causing damping off if you look above ground. And when we treat with our trianum, you see the roots look much better than the ones with only fusarium. So let's go to the registration. So our R&D department, our economical development department is really collaborating a lot with registration. There's a lot of interaction ongoing because we need to make sure that our R&D and also our efficacy trials are matching the registration requirements. So there's a few standard requirements as for plant protection products, and I'm focusing here on the plant protection products and not the biostimulants, because this is really the focus of Koppert. We really focus on the plant protection based products, so the microbes that are protecting against diseases and pests. So basically, already Andre also mentioned, they are still treated as chemicals. So we still need to comply to a lot of the chemical requirements. So one of the first requirements is the identity. So of course, when we have an interesting microbe, we need to know what the microbe is. And now the requirements are getting stricter, and they even ask now for an identity at the strain level. So not anymore at the species level, but they want us to provide markers at the strain level. And one of the other requirements, of course, is how do we produce it? How do we apply it? How often do we need to apply it? There are physical chemical properties that need to be investigated. And this includes, for example, the grain size, the color, how many carbon, nitrogen is in there. All those kind of properties need to be described and investigated. There's also analytics requested, and that involves secondary metabolites, proteins, for example, heavy metals. All those type of characteristics need to be analyzed, and the data need to be provided. Another requirement is our toxicity analyses. This involves, for example, toxicity like the skin, or when you get in touch on the skin or eyes or inhalation. It should be safe for everyone who's working with it. Also the residues, if our co-formulants are leaving residues behind, that all needs to be investigated. The environmental fate, also called E-fate. This describes the persistence in soils, sediments, and also natural waters. Then there's also the ecotoxicity. This is the toxicity, so the effect of our products on, for example, the bees, the birds, insects, and earthworms. And of course, there needs to be an efficacy also in the registration dossier. So for the European Union, it should cover at least 3 years of field trials per crop or crop group, per disease pest, per climatic zone. So you can already imagine that is, if you want to expand the markets, more field trials are required. And I can tell you this is a really expensive part for the registration dossier. Also, what you can see in the background is that there's different guidelines by taken by the European Union. There's the EPO guidelines to the EPO zones, but also the European zones. And you can already see that they are not completely the same. So for example, if we want to apply something in the northern region of France or the southern region of France, it means we cannot do like one field trial in France. It's two separate regions. So it is also making sure they're basically causing that we need to perform more field trials. So these standard requirements have to be done for active ingredients, so the microbe, but also for the final product. So, and we call it the 5-batch analyses. So 5 batches of the products, these analyses need to be done. We also have additional requirements, and that really depends on the type of application. So if you have a foliar application, you have different additional requirements than when it's a soil application. Per country, they can, even though it's the European Union, but each country can still ask for additional requirements. So for each country, we have separate registration dossiers. What is also quite interesting, or sometimes such a pretty hurdle, is that we don't know what will be asked. So sometimes we have a product and we basically cannot already expect what requirements they will ask for. And that is making it sometimes very tricky and interesting as well. So many of the requirements need to be fulfilled by GLP or GEP certified parties. So the general laboratory practice or general experimental practice parties, which are external parties which are performing all the experiments. So the estimated cost, for example, if we take a product, one product that is effective against 2 or 3 pathogens in 3 to 4 crops in all zones in Europe or worldwide, the costs are 3 to 5 million. And this is already indicating that this is quite a big investment. So we really need to take good care in for which pathogens, for which crops, which zones we want to register our product for. And the time frame is, as already described, is a very lengthy time. It takes 5 to 8 years before we get a biological registered. So what is then the importance for our R&D department? Well, the R&D department is playing a crucial role. So when we are selecting our microbes, of course, we play a role in the identification of the microbes. The identification, like I mentioned before, the requests are now that we need a strain level identification. Well, you can imagine that genome sequencing is really important there. And that's, for example, where we are collaborating with Keygene on. When we sequence genomes, then we can identify strain-specific markers. We use also that information for detection and quantification. We need to make sure in our quality control that our isolates that we have are still the same in the next coming years. And also, if we want to know how persistent our microbes are in the soil, how many times we need to apply, it's really valid to be able to detect and quantify our microbes when after application. In our microbial profiling department, we also are researching the activity spectrum of microbial actives. So this includes abiotic and biotic conditions. We need to know under which conditions our microbes are really effective. And this also, of course, can also influence our registration processes. If we know if a certain microbe is not so active in a certain crop, then of course there's no point of taking the effort of registration this microbe for this particular crop. We're also investigating the modes of action. So as Andre also described, most of the time, the microbes are having multifactorial modes of action, but we are investigating these to find out more how our microbes are working. And it helps us also to screen for new microbes with certain activities. And here also, for example, gene expression analyses are quite important. And that's something we're also collaborating with Keygene on. And we have regulatory restrictions, and we take that into account. And some of these regulatory restrictions are, for example, if we have a microbe that belongs to a species that could be potential human pathogenic, there's no point of trying to register it because it will be quite impossible. Also, the regulatory authorities are requesting whether our microbes are producing toxins. So if we know that already, we can exclude them from developing into a further product. For example, also for our bacterial products, the antibiotic resistance is quite an important requirement. If we have a microbe that has an antibiotic resistance and these antimicrobial resistance genes are located on mobile genetic elements, then I can tell you we would not proceed further in selecting this isolate for further processing, because it will be really difficult to register this product for our use. So our processing team is really busy with optimizing production procedures, and at Koppert we try to make products in a most sustainable way. And of course, we will still like to have increased yields and also good shelf lives. There are sometimes changes in regulations occurring. For example, that one of our raw materials is not considered as organic, and Koppert wants to produce most of our products as really organic products. And if there's a change in one of the raw materials, then we need to change our production procedures. But we have to keep in mind that this is also affecting the effectivity. So it might change the effectivity of our products, but also in registration. If it's a minor change, then it doesn't influence our registration too much. But if it's a major change, then we need to redo all our field trials and toxicity trials and all the other procedures that I just described previously. So basically, I think the R&D departments are really important also for the registration of the microbes. So the R&D is important in the selection of new microbial actives and to ensure that we produce actives that are good enough for registration and go quickly through the registration process. The process development is important to make sure we produce the microbial actives in a sustainable way and also in a way that the registration authorities can approve. So very important is that we're performing a lot of pre-trials of our prototypes to make sure we have a good quality, so that they are very efficacious, that they are safe, and really also reduce the time for the registration requirements. And these are basically the take-home message that I would like to give to you. And with that, I would like to thank you for your interest. And if you have any questions, please let me know.
Thanks, Irene. It's impressive how, articulated are the registration requirements. Thanks for illustrating us today this. So, maybe there are a few questions from the audience.
Yeah, there's a question from Prabha Karan. Can you specify the shelf life of Trianum?
So I think now on the product, it's a shelf life with storage at cold temperatures, so at 4 degrees for at least over a year.
Okay. And there's a related question from Jafar for Irene. Do you measure the post-harvest soil persistence of your products for compliance purposes? For instance, if you do that, how long can the Trianum P stay in the soil after the crops are harvested?
We are looking into that. I can't tell you the answer yet, but we are looking into that. Indeed. Yeah.
Okay. And a question from Han, which is more effective, foliar or soil application?
That depends on the product.
Yeah, I think so. I think it relates to the Trianum product or not? Can you say something from that on that?
So now Trianum is mostly applied as a soil product, but we are also testing whether we can use it as more as foliar products.
Okay. Question from Raju for Irene. What is the stable CFU of your applied microbe in rhizosphere over the crop growth cycle? Any studies conducted? Question mark.
Well, we are working on that as well. That's
okay.
So it's related to one of the previous questions.
Yeah.
That's so first we need to develop strain-specific probes that we also are sure that we are detecting our strains, because there's a lot of trichoderma, for example, in the soil, and it's that's our biggest challenge now is can we really trace back our isolate, and when we using probes, are we really finding our isolates instead of also other trichodermas? But we are looking into that. We are developing that.
Okay, there's a question.
Sorry.
There's a cat coming in.
That's the challenge of working at home.
So a question from Abdul Aziz. Microbial products efficacy is sensitive to many field factors. Do you establish agreements with growers in the long term for testing your products in the field trials? Or do you have your own fields or both? Or how do you do that?
So we have consultants who collaborate closely to the farmers. So there are some farmers that indeed we are testing at their location, but we also have our CROs, which are testing our products on more commercial fields.
So you are also a scientist who has been working in academia. Now you're working, of course, at the company, and we have seen your work in relation to the regulatory process and the development process. In your perspective, how do the R&D activities change while being in academia, or as you are now, while working in the private industry? Is there a lot of differences, or?
Well, actually, a lot of the work itself is very, very comparable as in academia as in industry. I think for me, the biggest surprises were indeed the registration requirements, that there were so many hurdles actually there. And also the time limitation. In the company is most often you don't have like years and years to investigate in depth how things are working. And in the industry, for me, it feels we need to have solutions quicker. And I think therefore the collaboration with the academia is quite important. And I think that can really strengthen, and that's also what André is saying, it can strengthen also the commercial businesses.
An interesting question, I think, from TZ. How do you see the possibility of the use of gene editing on microbial organisms to increase the yield or to remove toxins, for instance, to speed up the regulatory processes? And he or she, I don't know, but he asks it for markets outside EU at the moment. So it's a technical question, technical opportunity.
Yes. Well, of course, because if it would be within the EU, it's currently not feasible yet. There's, of course, a big debate ongoing on the gene editing techniques. Yes, I think it can benefit. But we also know that there's still a lot of diversity still present in nature surrounding us. So I think there's also a lot of more things in nature to explore before going to the gene editing. But that's, of course, something we also pay attention to.
And question from Marianne. So lots of questions. What is the difference of working in R&D between India and the Netherlands? Do you have experience in that?
I don't know. I don't have any experience of working in R&D in India.
So do you have any experience
I don't, unfortunately.
Not yet. Not yet.
That remains an open question, I think.
Yeah,
maybe...
I would like to know where the question is coming from, because they probably are hinting to something.
Well, I think Marianne is a Dutch name. Maybe it comes from somebody from the Netherlands, but...
Yeah. We'll see.
Question from Jill. What is your opinion on the possibility of breeding to increase the efficiency of plants to interact with the microbiome? So it's probably breeding better plants for better interactions. What is the possibility of that?
I think that would be really interesting to collaborate on that. That we can find plants that can attract the right microbes that can protect against the diseases.
And then sell the combination of the seed and the microbe, of course. Eh?
Yes, of course.
Yeah. Okay.
But it's the same as André was also saying. I think we also are looking into using biologicals together with chemicals to at least reduce the amount of chemicals that are currently being applied. That's because we also know that microbes are not always completely stable. And maybe not as effective, but at least if we can already help by reducing the amount of chemicals now being applied, it's already very positive.
So some questions, last question before we go to the general question and answers from Jafar. What is the level of root colonization of Trianum products?
We know it's colonizing the roots very well. but I cannot give any number, because also that depends on the crop. That depends even on cultivars. So it's not that we can pinpoint like one number to it, but we do see like a good colonization of our Trianum on the roots.
So thanks a lot, you all, the speakers, for bringing here your experience. And so I think it's time to move to the Q&A session. And I would like to start with a question for Maurizio, our biological scientist expert at Keygene. So we have seen examples on the impact that R&D has all across the company, starting from early choices in screening in order to accomplish or not to having many problems with the registration requirements, moving towards developing a deeper understanding of the mode of action of the product, whether it's for, for marketing and sales reasons or for building business relationships. So how do you think this could be more value added to these R&D trajectories?
Thanks for your question, Mireia. I think if there is one take-home message that we can have from today, it's that, to paraphrase Irene, R&D should be the backbone of what we are doing. And I think that's actually the only way of us turning this mentality, because I think in the past there has been a lot of negative, outcomes associated to biologicals, especially because products were not up to standard and they didn't deliver what they promised, because basically they didn't have what they were saying that they had in the label, right? So on the label. So I think if we take R&D as a backbone and then we invest in it and we understand what we are trying to accomplish and what the product does, that is a way of convincing ultimately the farmers, which will be the ones using. And I think that's how companies can benefit, can add value to their biological portfolio, because that is a trend that I don't think it's going to go away. It's the other way around. It's actually just going to increase. Yeah. And I think on top of, of this mentality that R&D can support that development, I think with the three examples that we had today, we can say that Keygene has the expertise and the capabilities to support companies in the ag biological field sector to, develop their products better, to deal with the challenges that they have in all the steps of their product development.
Yeah. Thanks, many thanks. So maybe we can take another questions from the audience. So would you like to read them, Arjen?
Yeah, I think in line with the previous question from Gabi, I think there is a question from the audience. Would an RNA spray targeted against an insect pest be regarded as a biological? I think it's a question for all of us who are now presenting. So who wants to answer this question? Irene or?
Experience in RNA as a alternative.
So an RNA?
I think now it's not considered as a biological. I think now it's more considered as a it's not like a chemical, but I'm looking for the word. Sorry, but it's not considered as a biological now.
Okay, so there's still chances there.
Any other views on this? No?
Okay.
Anonymous question for Martijn. Do you know if your Farrow product could be used to biofortified food crops? And what is the cost?
To biofortify, and somebody has to explain to me what biofortify means.
Yeah, so I guess it means to strengthen the plant resistance to some maybe abiotic and biotic stresses.
Okay.
I mean,
we suspect that the Farrow contains a little bit of fulvic acid. We know that fulvic acid is acting as a biostimulant. So yes, we expect that might be the case.
No, thanks.
But we need further proof for that.
Okay, it's never ending, R&D activities, but that's the beauty of it. So maybe a question?
Yeah, Ashok for André. Is it feasible to use Lumidapt, so root improvement, and insecticidal seed treatment together on a seed?
Yes, definitely. And we already have, I would say, plenty of data on not only with insecticides, but with the full pack. We have today Lumidapt being used together with fungicides, insecticides, nematicides, all together in the seed, or standalone, or only with fungicides, or only with insecticides. So over these 10 million hectares that I was explaining earlier today, we already have all those mixtures, and yes, it works very well.
So I will pick one question for Arjen, because, you know, it's nice that you read questions for yourself. But what is the main conditions to collaborate in the R&D fields?
Yeah, in general, I think in this type of work, I think it's trust and perseverance. It's not easy to develop agricultural products, being it seeds, new seed varieties, or being a new biologicals. It always requires a lot of efforts. It requires a lot of time. So I think trust and perseverance, which is then depicted in a solid contract and agreements around investments and revenues, but also who owns the results. And IP is a very important one. And so that is very important, I think, in this type of research. There's nothing like a quick fix and a quick win, I think, in our business. It always comes back to big efforts and sometimes lengthy efforts.
Yeah. Thanks. So there's a comment that about the snake oil products in microbial biostimulant market. So of course the regulation is coming. So I guess Irene and the others have commented also what is the role of the regulatory requirements, not only to make our life more difficult, but also they have also a really important role that is to provide efficacy, sanitary products, and so on. So thanks. So maybe we can read another question.
So Noor for Irene, do you feel the increasingly strict regulations? And I heard that Corteva was even saying, well, we are doing the same type of regulatory investigations for our biologicals compared to chemicals. But do you think that the strict regulations for your products are hampering transition towards sustainable agriculture?
Yes. No, I think we have to keep in mind that all these requirements are really to ensure safety, and of course that's also very important to us. But I think some of the requests that are now being asked are sometimes going beyond feasibility also. As an example, they are requesting that we are investigating all the metabolites of the complete Trichoderma genus, whether our strain is producing all the metabolites, which you can imagine is like not feasible to study, especially because for many of the metabolites, gene clusters are unknown yet. So it's not that we can screen in the genome sequence for these pathways. And it's also really tedious to screen all metabolites that are now being identified for the whole complete Trichoderma genus. So I think it is definitely hampering there, that we need to deal with those requests in the best way that we can, and to make sure it is feasible. Because as you can see, it's also a really huge investment for the registration requirement. We have to make really clear decisions of where we want to market our products.
Question from Christophe to André. Do you see positive effects of Lumidapt on the next crop in the field? So it's 2 for the price of 1. What is your answer on that?
I think we do not have studies on that, but based on the amount of product that we put, and this is really a small amount of product, I don't expect that we're going to have this long last effect. even for the crop, Lumidapt is used during the seed treatment. So we aim to provide to the crop the better crop establishment. So crop establishment meaning the first 2 weeks of the crop in the field. So even during the same cycle, we recommend, in case of needed, other foliar applications of biostimulants. So I don't expect that in the season after to the other crop, we will still have, I would say, significant benefit in the ground. So
we want to come back to the question about the R&D differences between or similarities between India and the Netherlands. And the person who asked the question said, well, I asked the question because Koppert is not only present with R&D in the Netherlands, but they also have an R&D site apparently in Bangalore. So does that ring a bell to you, Irene, or not?
Yes, well, that's probably more the economical development. Then that's sometimes also put into the same field as R&D, because the real R&D departments are mainly based in the Netherlands, in Brazil, and in Spain.
Okay.
Okay.
So it's maybe due to the field trials and testing.
Okay, so I will rephrase another question for you, Arjen. So what is the percentage of R&D budget, of the budget that Keygene dedicates to R&D?
Well, Keygene is a pure R&D company, so my first reaction would be that probably all of our activities are R&D, maybe with some exceptions with license income on our patents, but the rest of our turnover is all on R&D projects. And basically, we have three types of projects. Projects we run from our own internal resources, projects that we run with our strategic shareholders in the vegetable and fruit industry, and projects that we run with field crop companies, biological companies, agro-food companies, and that's basically all our activities.
Yeah. Yeah. So many thanks. We have arrived to the end. Thanks to the speakers for being us and sharing your experience today with us. So many thanks. I hope you, audience, have enjoyed the webinar. It has been registered, so whenever you want to review it, or for those who couldn't attend, we will share soon a link to access. The questions have been saved, so we will take care of addressing them shortly to the relative speakers. And again, thanks for being here today. There are many more examples at our webpage, keygene.com/biologicals. Please contact me whenever you would like to discuss possibilities for R&D impacting your company. And well, have a nice day, you all. See you soon.
Bye-bye.